Hi everyone. Welcome to this week's NCT Data Science Seminar. And thank you already for organizing all of this, Dorin. Usually Lena's hosting this, but she's not here today.
She's online.
She's online. Okay. Hi, Lena. And so it's my pleasure to introduce Zdravko Marinov, a PhD student from KIT. And I met Zdravko around 2 years ago, I think. We started working together on a review paper on interactive segmentation, which was recently published in TEPAMI. And I remember from the first meeting on, I was very impressed with this back then second-year PhD student, how well organized he was and how already on point you were discussing and so on. So I already predicted back then that you're going to have a great future in science. And I think today we're going to see that this is going really well. I'm really excited, especially also because your topic, interactive segmentation, is very similar to the name of my group and my research. So yeah, we're really excited to have you here and looking forward to what you're going to tell us.
Thanks for the kind words and welcome everyone to my presentation today. As Paul mentioned, this topic is quite interesting for his group and it really aligns with what they're doing there. And it's getting a lot of traction in the medical image analysis community in the last years. But I really want to introduce myself first. I'm a PhD student at KIT. I did my bachelor's and master's there in computer science. And right now I'm under the supervision of Professor Rainer Stiefelhagen at the Computer Vision for Human-Computer Interaction Lab. And I'm also part of the HIT for Health graduate school. And my topic is exactly the topic of the presentation today, which fits perfectly. I am a contributor to MonILabel, an open source project exactly for interactive segmentation of medical images. And we have a strong collaboration with the University Clinic of Essen and Institute of AI with Professor Jens Pesek. To go into the topic, I really want to motivate with an example from the natural images domain. Let's say we look at the cityscape dataset and how it's annotated. An image from cityscape datasets is around 1,000 by 2,000 pixels, and the paper said it's around 90 minutes to annotate a single image. And there's 5,000 training and test images to annotate in total. So a wild guess how much time, when you crunch up the numbers, that will take to annotate the whole dataset. The answer is actually around 312 days non-stop, without sleeping, without anything, without eating, or for workish if you consider 8-hour work days. And that's quite a lot, quite unfeasible when you think about it. Do we really need to click all of these pixels and manually annotate the image? The answer was yes. We wouldn't really have this presentation today. So the answer is no, and most of the commercial software for annotation and open source software is actually interactive segmentation of these images. And this reduces the amount of effort you need to provide to segment such images. Current state-of-the-art approaches like this one could segment an object using just 2 to 3 clicks. This reduces the annotation time significantly. But of course, today we want to talk about medical images. So let's give the same example for the medical domain. In this case, there's some differences, but interactive segmentation methods are really generalizable. So you can apply the same principles you do in natural images to the medical images domain as well. So let's take a look at the example from the AutoPet dataset. It's a PET-CT whole body FDG dataset with lesions. Here, the difference is the annotator must have a medical expertise to annotate these images, and he needs to understand the anatomy or pathology we're talking about. And also, the complexity of the data is much larger. You have volumetric data, you have diverse modalities, diverse patients, diverse scanning protocols. The diversity is much, much larger. And it takes 1 hour to annotate a single volume, just with one class, just the lesions. And there's around 1,000 training and test volumes. Again, we have a lot of time dedicated to just annotate these images. Here it comes up to 3 work months, but you can think about that the medical expert is quite expensive and it's difficult to organize such an annotation study. So what we actually did, while to load, So what we actually did in last MICI submission publication is to have this collaboration with the University of Medicine Essen, and we brought down in interactive segmentation the annotation time of the whole data set to just 10 clicks or just 1 minute per annotation. We organized an annotation round with the radiologists and medical students from the clinic of Essen, and we managed to annotate the data set in just 2 work days to some amount of I-score, around 90%. That leads to a very much more efficient way to annotate such images and much more quality control once you refine the segmentation by adding new interactions. But before we go into details and research highlights from the recent years, I want to define what exactly is interactive segmentation. Let's say we know what non-interactive segmentation is. We just have this black box segmentation model. It could be a CNN, it could be a transformer, it could be even k-means clustering. We have just one step in the segmentation process. Let's say it's just one inference step. And then you have the segmentation output. In interactive segmentation, you bring the person or the annotator into the prediction process by having this iterative feedback loop, by adding interactions in the process. For example, you can initialize segmentation with 2 clicks and segment the cat's head. But then you want to segment really the whole cat, so you refine the process by adding more and more interactions throughout the process. And this leads to implicit quality control of the labels. You can add clicks wherever you think the model has made a mistake. That's really important for the medical image analysis because we have this implicit control of the reliability of the model. And we also update the predictions in each interaction. There's different dimensions to designing an interactive segmentation system, and I really want to go through each one by one to show what exactly it's about. Starting off, when you start designing interactive segmentation, technical issues here. Yeah. When you start designing an interactive segmentation model, you have to think about first what type of interaction you're going to use. For example, clicks are very intuitive. You can just add a click in the object you want to segment, or add multiple clicks to annotate it. For example, this included sheep here, or add a background click if it's over-segmented. But although they're very simple, they're also quite ambiguous. For example, if you have this example with a boy, you click on the boy's pants. It's not sure whether you want to segment only the pants, the boy, or the sitting boy. So you need to provide some additional information to the model, such as a text prompt, a sitting boy. Scribbles add another layer of complexity on top. It's like a set of clicks, and then you can be more specific with the object you want to segment. For example, the whole cat, or add a background scribble on its body to specify that you only want the head. Another popular approach is just using boundary polygons. A very popular approach in commercial software where you segment with one model and then manually correct the vertices of the boundaries of that. Or bounding box where you just segment through the bounding box. Then the next component I want to show is the guidance signal. That's terminology from the interactive segmentation domain. So you first have to define it. The idea is that once you choose the type of interaction you're going to use the model with, you have to think about how you're going to implement that into the model, how you're going to provide information to the segmentation model. So let's say we get clicks for our segmentation. How do we present that to the model? There's quite a lot of different ways to do that, to implement it. One is to explicitly define an additional input to the model, such as goals and heat maps or constant radius disks. You can also define distance transforms, such as Euclidean or geodesic distances, and many more. These are one of the most popular ones, which I've shown on the screen. And then you algorithmically or deterministically compute that from each click. Then, for scribbles, another popular example is to skeletonize the ground truth mask. Here, you just compute the medial axis transform, and to make it more realistic, you can perturb it in a way by sampling it and warping it, because you're not going to expect clinicians to know exactly where the skeleton of the ground truth mask is. And there's also a possibility to integrate interactions implicitly as a guidance signal, so you don't really have a thing, but more of an action in the training process. A very simple example is to weight the loss function during training more in when it's near the interactions. So, for example, if it makes a mistake near the click, you have a higher loss function, and then you implicitly make the model listen to the interactions. or to pre-process the inputs in a way with bounding boxes you can crop the image, and then you have this localized context which you work with. That's sort of an implicit action you take to process the image. To take away guidance signals or things or actions that actually make it possible to work with user interactions and introduce them to the model. So is the guidance signal that important? Is it just another hyperparameter, or is it something which we need to think about more carefully? The answer is that in our previous MICI publication from last year, we analyzed popular guidance signals, the ones I showed earlier, heat maps, disks, and distance-based transforms. And we saw a trend that a lot of the signals have two pitfalls. The first three signals here are very localized. Each one of the clicks conveys the same information. It's the same for each one of the clicks. So you cannot really differentiate where the location of the click is. So if it's near edges or if it's near homogeneous regions, it doesn't really matter. It's position invariant. Whereas distance-based transforms, such as the geodesic distances, are convey this information, but they're really noisy. They're really difficult to work with because you have to define thresholds where you cut off high distances and low distances. And this leads to these two pitfalls. And the thing we wanted to design is something that combines the advantages of both signals. You want to detect edges and you want to make it more precise near very fine details. This is exactly our adaptive heat maps approach, which managed to outperform the other guidance signals. The way it works is that you compute the localized geodesic distance around each click, which computes the geodesic values that convey the information where you actually have edges in the image. That's a simple way to say it. And the way we map these values to click is with this negative exponential term. I'm not going to go into too much detail in the equation, but you can imagine that if something happens in the image in this local context, it's much better to be careful because you have edges and you can map that to a lower radius here. But if you're in a safe region where nothing really happens in the geodesic distance, then you're probably in a homogeneous space where you can be more liberal with your size of the heat map. This leads exactly to this mapping here. So all in all, if you have more details, you should be more precise and have a smaller radius, and vice versa, fewer details when you have a larger radius. And since we learned our lesson from the metrics for rolling paper, never use just one metric, don't just report the die score. We actually designed five metrics, and explored the parameter space here compared to the other guidance signals. So we looked at the final dice, the dice after 10 clicks, the initial dice, so that's the dice before adding 10 clicks, because when you train an interactive model, you kind of bias it to listen to clicks, and the initial interaction might deteriorate, so the initial prediction. And if it's a higher dice score here, then you have a lot less work to deal with during annotation. We also looked at the efficiency when you do inferencing into the guidance signal as well. And we defined two new metrics. One is this called consistent improvement. That's the ratio of clicks that actually do improve the performance when you infer with the model. That actually is not always 100% because it's an additional input, and neural networks are not really deterministic. So you cannot expect by adding a new click to have a better performance. And the ground truth overlap. So how well does the guidance signal align with the ground truth mask? So in the table before, I show that we do have a better dice score, but you can see it's not that significant. It is better, but what we want to look at here is the usability of the model. So here we see a very large significant improvement in these two metrics. That means that a lot of the clicks will be much more useful for the annotators because you have a more consistent improvement of the segmentation, but you also have a much more aligned guidance signal with what you want to segment. This is seen in both the dataset MSD spleen, which is this more simple variant, and Altpad, which you have this much lower dice scores. So as a takeaway, guidance signals do matter, and it's a very important design decision when you think about interactive segmentation models. This is exactly the thing that brought us to 1 minute annotation of PET-CT volumes, such as showed earlier slide. The next dimension is called robot user. Again, something from the terminology of interactive segmentation models, so it's not really popular as a term. What it actually means is, going back to the example of the cityscape dataset, let's say we already have this model and we want to annotate the 5,000 images from cityscapes. This would be called the application phase of the model because we're annotating new data. So where did we actually come up with this model? How did we train this? The ugly truth is that you need an even larger annotated dataset to annotate the smaller dataset. So that means you need to pre-train the model first. And for this, you need, let's say, 100,000 images. How would you create interactions for that? Would you ask an annotator to click each one? Even if you do only one click per card in the cityscapes dataset, then you would have 100,000 clicks. And that's really a lot and still quite expensive. So the motivation behind the robot user is to simulate these clicks. And then you also don't have the limitation to only have just one click. You can simulate multiple clicks per object. And that leads to the simulation of the interactions during training. Robot users are not just limited to training. Later, I'll also show you how you can apply them during evaluation to save your a bit of time for annotation studies. So the blueprint for an interactive segmentation training process is given here with the robot user. It has 4 steps, and going through them one by one with the earlier example of the CT image for screen segmentation, we can see that it's really quite simple. The first step is just the initialization of the interaction process. So you have to start off with a simple click. You look at the label, you look at the image, and then you simulate a click that is valid within the label. So, for example, this, and then you transform it to a guidance signal so you can process it with your segmentation model. You give it with the image, you predict, you have something which is not perfect. So then you enter this iterative refinement loop, and this is the key to the interactive segmentation process. Because in this case, you can look at the label, you can look at the prediction, and then correct this and rectify the wrong areas. So you add a prediction, you look at the prediction and add a guidance signal again in the area where the model has made a mistake. You repeat this again, you look at the image, you look at the guidance signal, you predict, you compare it with a label, the robot user adds a click somewhere in the segmented area. And in the end, you define a stopping criterion. For example, let's stop at 3 iterations for each image during training. And we backpropagate for a model. Usually, you do this backpropagation at the end. So you actually accumulate the interactions in the guidance signal here, and then backpropagate. There are some methods, but it's rarely seen where you can backpropagate here, where it's in the second step. But it's much more computationally expensive to do that. And to train the model, you just repeat this whole process, this whole blueprint for each image in the dataset for multiple epochs. And then you have this large segmentation model, large training scheme for this segmentation. So a good question here is, how does the robot user decide where to put a click, in the label or in the segmented area? A lot of the methods we did in this systematic review, which Paul mentioned, are using the center click. So you just use the centroid to the label or the centroid to the error, or maybe some uniform random sampling works as well. But is this actually realistic behavior? Is this something which annotators actually do? And to understand that much better, we have to think about how we evaluate interactive segmentation models. There's 2 routes to do this. One is to really do this annotation study with real medical experts. For this, you need to set aside time and you need to organize everything. And it's really difficult because you can only afford a small sample size usually and a few annotators. But what actually you report in the end is the real performance of the algorithm and the real performance of the model. And that's really useful to understand how well the segmentation model works. The other route on the right is you simulate the interactions also during evaluation. So you don't not only do that during training, but also evaluate with a simulated robot user. Usually you can use just the whole test split of your dataset or the whole dedicated test dataset, which we have set apart. And this could be quite large since it's simulated. And most current approaches actually will go down this route because it's much more easy. Again, let's ask the question, is this realistic? Will the results be the same as if you would do them in the user study with real annotators? And we have to think about pitfalls when using such simple robot users. Just taking the centroid or just doing random sampling does not work so well because you have this very perfect robot user. It's something that always simulates valid clicks with respect to the ground truth. But you have to think about medical tasks that there's a lot of label inter-annotator disagreement. Because when you have a very difficult task, there might be a lot of different opinions on it. There's always the discussion board, whether this voxel is part of the tumor or not, whether we should consider this target or not. And you have to think about that depending on the task. And the robot user should not be the same for all tasks. It's also too deterministic. Simulations follow this blueprint, these predetermined steps. while real interactions are much more variable. You might have an annotator that prefers to annotate the center of the lesion, for example, and an annotator that goes around the boundary of the lesion. So this is something that you need to discuss with your annotators before you evaluate on a large scale with the robot user. And our publication in MICAI this year, which I will present next week in Marrakesh, goes exactly into this route. So we took a look again, like similar to the guidance signal study, we took a look at popular robot users used in literature, and we compared them. So we took a look at 4 popular robot users, one that uses the center click, one that simulates a click in the encircling regions, one that uses the Euclidean distance transform, and one that uses uniformly samples random clicks. We also conducted 2 independent user studies with 8 medical annotators from various backgrounds, from medical students to radiologists and to a nuclear medicine expert with 4 years of experience of annotation. So we have a lot of diversity there. And we did this in collaboration with the University of Essen, again with the Institute of AI for Medicine. The dataset was autopets, which I mentioned earlier. We used 20 models for that. What you would actually see here is a very evident gap between the lines on top and the lines on the bottom. What you actually see in these colorful lines, like these 4 colors there, is what you would report with your on-the-shelf interactive segmentation model. Let's say you train something on autopet and then report with a robot user. And then you see these really nice numbers and you publish a paper and you say, yeah, our method is state of the art, it's so great. And it's awesome, just look at the numbers, we need everything else on the list. But actually, what you would report if you use real annotators, in 2 independent user studies we evaluated this, is much worse and much more realistic. So what we actually report with such simple models that are too perfect and too deterministic leads to these pitfalls and this really bad results, really large shifts in results. So the last question is actually, how did we design a robot user that fits the behavior of real annotators so much better than what's out there? So as I said, we conducted 2 independent user studies. The truth is, the first user study, we didn't plan on implementing any robot user. We just wanted to experiment with what kind of behavior we would see. So we took a look at the annotation after we saw this gap. And we saw that there's a very large disagreement between the annotators and the annotated data. The disagreement of the ground truth came in 2 forms. One was this very close clicks to the lesions. So that would be something like you don't agree on the boundary of the lesion. And that's really, really specific on the type of software you use to annotate. For example, if you use 3D Slicer, you might change the window link in a way that it looks ambiguous where actually the lesion ends. And we saw a lot of these errors, but we also saw a lot of disagreement of what is actually a lesion. So you have these clicks that are really far away from the lesions that are annotated in autopets. And this really surprised us because this label non-conformity came in 25% of the clicks in the ground truth. So this is a very, very large disagreement with the ground truth. And simulating everything perfectly did not work so well. Again, we went down the path of designing metrics to evaluate everything. And we also used 5 metrics this time. We reused the consistent improvement metric from the guidance signal paper. And we designed 4 new metrics to estimate the annotator behavior. So one of them is exactly this label conformity. How many of the clicks actually are within the ground truth label and how many disagree. And so this is this proportion here, M1. We also looked at how annotators click, whether they click more near the center or more near the boundaries of lesions. We estimated the distances of between the centroids of each connected component. We also looked at the diversity, so that's another metric to estimate behavior. If you have, for example, 10 lesions and 10 clicks, would you distribute the clicks in 10 lesions or would you concentrate to making the first lesion perfect? So that's what this estimates here. And we use the label proximity for label disagreeing clicks to estimate what's the proportion of the first case where you disagree with the boundary and the second case of nonconformity where you have exactly new lesions. So this is the distance to the ground truth of nonconforming clicks. And we wanted to estimate everything at once. So this is the concept of a user shift. In essence, it means how much does the annotator shift from training to evaluation. In this case, you have 2 types of shift. You have this training with simulated interactions and shift to the robot user, which is also simulated. but you can also estimate the shift from simulated and real interactions from the annotation study. And we use just the mean absolute error or just mean absolute difference between these. And we try to model these types of errors, which I mentioned at first. The label nonconformity in the terms of boundary could be modeled by just perturbing the click with a certain amplitude, a number of voxels. And we try different percentages of clicks. For example, how often do you have this type of disagreement during the evaluation process? And we actually saw that the optimal way to optimize this user shift is to have this orange curve here, which corresponds exactly to the 25% disagreement. And interestingly, we conducted the same thing for the second type of disagreement, where we just sampled clicks in high uptake regions, high metabolic activity regions in the PET image, but which are not in the ground truth mass, so something which should be disagreeing with the clicks. And we also had this 25% disagreement, the optimal way to optimize the user shift. So we modeled our adaptive heat maps to make this type of disagreement 25% of the time. So we had this joint distribution. We tried 25-0. We also tried equally distributed, and that worked the best. And that not only led to the best user shift, to the least user shifts throughout training and validation, but also the least dice differences we saw in the curves here. So that leads to much more realistic evaluation, and we should really think about how we evaluate such methods, because it's something which is a really common practice to use the center click fault user and just be done with it. And the last dimension I really want to discuss in the interactive domain before heading throughout what's happening in the field right now is active learning. It's just a very short dive into it. But active learning is really coupled with the interactive segmentation because it lets you select the most informative samples to annotate. It could be done in two ways, and that one is to annotate new data. For example, you have this unannotated data set. You present it to annotators, they annotate some of the samples. You train a model, you predict on the whole data, then select the most informative ones. The annotators would correct that, and then you repeat this whole process until you have a model that is pre-trained in such a way that it's more confident in its prediction. Then you use it to predict on the whole unlabeled data and use the pseudo labels for downstream training. So you can say the end result is annotated data. The second way to implement it into interactive segmentation is to refine a model. For example, when you task on unseen data, so it's already pre-trained on some other task. You apply it to the unseen data, you predict on it. It's not going to be perfect, so the refiners will add more and more interactions to make the predictions good. For example, by adding a guidance signal to guide the model to predict much better on this unseen data. And then you actually have, after a few steps, a model that is adapted to the new task, and you can use its prediction for a more accurate estimation. There's different ways to select these most informative samples in the two steps. I don't want to go too much into detail here, but as you know, we can use ensembles, different models from different checkpoints, for example. Monte Carlo dropout, the same model, but you drop different types of weights, and then you look at the disagreements between these models. You can also augment the inputs, or you can even dedicate a model to estimate the uncertainty. It's a bit more niche, but there is some papers which actually do that. So after discussing all of these dimensions, I want to really dive into what's happening in the field right now. And for this, as Paul mentioned, we had this collaboration between our lab and the DKFZ and also the University of Medicine Essen to conduct this systematic review of what's actually happening. What we did is we used the keywords deep interactive segmentation medical, and we searched through Google Scholar and PubMed in the search engines. We added some seed studies which fit our criterion. Then we went through this tedious loop here. First, we went through connected papers. We looked at the papers which actually do fit within our scope. Then we looked at the papers which have cited these papers and their citations as well. Then we looked at all of the different venues that the papers have published in and looked at each one of the papers one by one. Of course, we limited 2016 to 2023 because it would take an eternity to finish this loop. And we looped around, there were no more relevant papers. And that led to this final paper stack, which actually landed around 121 papers in the systematic review. But we didn't really just want to sample the papers and summarize them. We wanted to organize them in a way that it's useful for the community. So we set out to create a taxonomy tree of everything that we've sampled. And the question was, which taxonomy features could we use? So we mentioned these 4 things, these 4 dimensions when designing the interactive segmentation models, but they did not really fit within splitting the tree into distinct features. For example, you can use different types of interactions, and you can have something that uses bounding box endpoints, so you cannot have this clear separation. A guidance signal is also implementation specific, so you can use an ablation study with the same model with different guidance signals, so you cannot really split that. You don't need simulations to create an interactive segmentation model. As I showed in the active learning example, you can just use real annotators. You don't have to simulate everything. And the same goes for active learning. You can use simulations and not real annotators. So what is the good split here? How do we split the papers? As I mentioned earlier, there are 2 phases of interactive segmentation. One is training and one is application. So during the training, you actually optimize the model and make it ready to annotate or to refine on a certain task. And during application, you actually apply it on the final data you want to use in the clinical setting. So we separated all of these works with these 2 stages, training, application, and hybrid methods called online learning. We designed the taxonomy tree in such a way that you ask questions at each split. So if you're someone reading the paper and you want to design an interactive segmentation model, you can go down the tree, ask these questions for yourself, and land on these papers at the end. This gives you some overview of what exactly you're going to do, similar approaches, and it also eliminates the excuse that there's no related work to compare to. So you have to compare to these methods if they make it publicly available. The different stages here, we also summarized each one of these in a taxonomy blueprint. So you can imagine that each one of them has its own blueprint. The one I discussed earlier was simulated iterative simulation during training. You can see that the human annotator is involved only during application or during training in these different blueprints, except for the online learning, which is hybrid. And it also summarizes how exactly everything is implemented, so you can follow them really strictly and really in detail. We saw a few positive trends here. As I said, it's an emerging field and a lot more is happening each new year. I can imagine in 2024, we stopped the scope in 2023. It's even larger due to SAM and MedSAM going into the field. But there's also a positive trend in terms of how many of the works actually use public code, how many of them just use private dataset, and everything is looking really nice in the field. So we can expect a much more reproducibility here, right? So to answer that, I think you know where I'm going. There's a lot of chaos in the field. You can see this large graph here. That's methods from the systematic review that you have compared to each other with the arrows. And there's no really systematic way to look at it. It's really chaotic. But the real ugly truth is that really on the bottom. So you can see that 67 out of all of the reviewed methods did not compare to anything from the interactive segmentation domain. And that's a really concerning trend, and we really need to understand why that happens. Another concerning thing is the evaluation metrics which people use. It's quite scattered, it's not really established in any way. But the most concerning thing is that we're talking about interactive segmentation, and interactive metrics are really underrepresented here. For example, very few works are reporting how much time it takes during the interaction process, or for example, the dice score on a certain number of clicks or other. The most represented one is just the dice score, and that's really not going too far into the interactive domain. What exactly is the problem here? Why do we see this? The answer could be summarized that there's no standardization for the comparisons. There's no established baselines here. There's not even go to at least compare to any unit. You should always compare to any unit with non-interactive methods. So there is no such thing in the interactive domain which you can point to and say, yeah, at least you can compare to this. You should start from this. There's no benchmarks, at least no established benchmarks. For example, the robot user is, the central robot user is used a lot, but people use different types of data set, they use different types of formulation protocols, and everything's really chaotic. And we know that these robot users are not realistic, and we need to think about it more carefully when we evaluate such methods. And there's also no established evaluation metrics. So everyone's using different metrics, we saw this diversity, and there's a really lack of interactive metrics. So there's really no standardization in this field. And now I really want to ask, are we asking too much? Is this too critical of the field? And To give a good example of success stories in other fields which are related, for example, interactive segmentation of natural images, not medical ones. We do have this established robot user. Again, they use the central click, but we will save that for we will save them that. At least they have established data sets and established metrics, and we have this really nice table which you fill new rows in. You know how it compares to previous work, and you know that it is somewhere building on top of each other. These protocols are also implemented in a very standardized way, and people can, authors can compute and report all these metrics themselves. Another success story is the Touchstone benchmark. It's a recent benchmark that goes into non-interactive segmentation of city images. You have this abdomen atlas data sets, you pre-train your model on, you submit it as a challenge submission, for example, to the organizers, and they have this really nice systematic way to evaluate the methods, establish metrics and establish data sets. And you can build on top of that to create something which is much more reasonable than this chaotic mess which we saw earlier. And the one which is most on point is the interactive segmentation of medical images challenge, CVPR competition hosted by the authors of the MedSam paper. So they actually released part of the training data set they pre-processed for MedSam, and the competition aimed to create a universal interactive segmentation model, which you can use to segment any modality, any structure. So it was, again, not exactly what we were looking for, but it is somewhere aiming for the right direction. Again, the results were computed centrally by the organizers. They actually used 3 metrics, not just one: the dice score, normalized surface distance, and the efficiency, which made a lot more sense than the metrics which we see during our systematic review. We actually even participated in that challenge, and we landed the first place in the scribble-based track, but more on that later. So what actually lies ahead? How do we fix all of the standardization issues which I mentioned? The baseline is one of the components we want to add to the interactive segmentation domain. We cannot really say we have an NN unit for this domain. MedSam is sort of paving the way for universal models. We also have Scribble prompt, we also have Uniseg and other universal models, but there is no expert models yet that adapt to that domain in an AutoML way or another way that will work in the interactive domain. And I'm saying this because many of the methods actually outperform MedSam in the challenge, which I mentioned earlier, which is a universal model. And most of them are mixture expert models, including our submission to the challenge. And that seems to be the better choice currently for interactive segmentation. We don't really have a benchmark for interactive segmentation. We saw these 2 success stories. We need to also expand it to various task modalities and think about what's actually suitable for a benchmark. Really like this publication, Nikai, that we're going to see next week in Marrakesh presented, and think about really good, in a really systematic way, what we want to represent for our benchmarks before building on top of it. Because once you establish a benchmark, you're really stuck with it for multiple years, and you really have to think about in advance about it. In the interactive domain, you really also have to think about this robot user. The way I see it is that we have to talk with the people that are using the models, exactly how the interactions are done, how they annotate the data, and what's important. And you can really not go around this human factor, because interactive segmentation is actually done for the people. So you need to talk with them and do interviews and go back and forth until you have something that works. And of course, we need interactive metrics reloaded. These are all really large claims, really ambitious things to say, but we really do need all of these things. And we need a systematic way to select the best metrics for a current task, going down this very nice structured way as metrics reloaded. And this should be, again, based on the task and modality, but also based on the way we annotate in that domain to fix this fragmented nature of these evaluation metrics. And of course, we cannot do this on its own. You don't have a benchmark without a metric. You don't have a benchmark without a baseline. Each one cannot really exist on its own. We need something that is combined, and that would be the interactive renaissance exactly for this domain. And with this ambitious task listed here on this slide, I would like to end my presentation, and I'm really happy to take any questions from you.